Hello everyone, my name is Esmeralda Escobar Muciño. Today I'm presenting the work titled Molecular Dynamic Classic Study for TDX and 6-GNG compounds as competitive inhibitors of LasR transcriptional regulator of quorum sensing in Pseudomonas aeruginosa. The quorum sensing mechanism regulates the transcription of phenotypes in Pseudomonas aeruginosa. By the other side, the inhibitors of quorum sensing are used as therapy against the virulence factor regulated by quorum sensing. The aim of this work was the screening of 1,500 aromatic compounds by molecular docking with LasR transcriptional regulator of quorum sensing and determine the best inhibitors molecules of quorum sensing in Pseudomonas aeruginosa for propose a new therapy against virulence of this pathogen. The methodology of this work is described below. 1,500 compounds from 6 family aromatic compounds were tested in a molecular docking screening with LasR transcriptional regulator of quorum sensing in Pseudomonas aeruginosa. The results were visualized in Chimera software as well as its ligands in Visual Studio software. Then the best results were tested by preclinical studies, while the molecular dynamics of protein ligand studies were performed to validate the molecular dockings. In addition, the total Gibbs energy was obtained to select the best in quorum sensing inhibitors and propose them as antivirulence therapy. In this picture are shown the results for molecular docking screening with protein regulator LasR and the 6 families of aromatic compound families demonstrated the Gibbs energy values between -11 to -14 kilocalories per mole. The best results of the molecular docking screening are shown here and were acceptable to Lipinski's rule of fives. The best protein-ligand interaction obtained from molecular docking screening were drawn and showed the complex of aromatic compounds in the autoinducer domain binding of transcriptional regulator LasR of Pseudomonas aeruginosa. The best results were validated by molecular dynamics in NumD software. The RSLD results are shown in this picture demonstrated the stability of all complexes. Then, the total Gibbs energy was obtained and 2 compounds were proposed as possible inhibitors of quorum sensing to be tested in the experimentation with animal models. The conclusion of this work were through molecular docking screening was possible to identify 2 compounds from 6 aromatic compounds families as possible quorum sensing inhibitors of Pseudomonas aeruginosa because these compounds block the autoinducer domain binding of LasR transcriptional regulator of quorum sensing. In addition, the molecular dynamics allow validated the molecular docking results. Finally, the Gibbs energy helped to understand how strongly the complex bound and permit to select the best quorum sensing inhibitors.